Well, hello and welcome everyone. My name is Nina Luning Prak. I'm the chair of the AIRR Community, and it is my great pleasure to introduce this meeting to all of you. This is a three-day virtual conference called Leveraging AIRR-Sequencing Data to Inform the Biology of COVID-19. So in this brief 10-minute overview, I just want to quickly describe for you what the AIRR Community is all about. And then I want to talk a little bit about what the AIRR Community is doing in the global effort to fight the pandemic, and then introduce for you very briefly our meeting here. So let's get started. So what is the AIRR Community? So AIRR stands for Adaptive Immune Receptor Repertoire, and AIRR-seq refers to the profiling of antibody and T-cell receptor gene rearrangements. And this occurs by next-generation sequencing. So the AIRR Community is an international community of over 200 labs and other stakeholders that are involved in AIRR-seq. So now I just wanted to give you a brief kind of historical overview of how the AIRR Community started. So in 2014 and 15, there were a series of meetings that were held, and the principles of standardizing and sharing data that drive the AIRR Community's efforts were first articulated by Jamie Scott, Tom Kepler, and Felix Braden. Then in 2016, the AIRR Community was named and formed working groups, and we began working together on a whole series of collaborative projects. In 2018, the AIRR Community joined the Antibody Society, and that's why you'll see in our slides here, we have both the AIRR Community logo as well as the Antibody Society's logo. The Antibody Society, or TABS for short, is an international nonprofit society that supports antibody-related research and development. So the structure of the AIRR Community, it consists of a series of 8 working groups and 4 subcommittees. So you can see the working groups there, and there's far more detail given about all of these working groups at our website that's listed below here. So the AIRR Community has these working groups which really collaborate on the development and adoption of standards and best practices for AIRR-seq data generation, analysis, storage, and sharing. Now the subcommittees, I really want to highlight the meetings committee for today's effort, which has members David Banyara, Pam Borkhardt, who is our meeting manager and source of all knowledge in the AIRR Community, Jean-Philippe Burkhardt, and Ramit Mir, and Jamie Scott. We also have a communications committee and a Fer de Lille committee and an executive subcommittee. So just to give you an illustration of how things work in the AIRR Community, we have a series of working groups that work on projects and then produce work products. So for example, the Minimal Standards Working Group developed data sharing standards for not only how you share data, but also metadata, the data that describe an experiment, for studies involving AIRR-seq data. And here's a paper that came out in 2017 from that. We also have a data representation working group, for example, that has developed file formats and specifications for sharing of AIRR-seq data. And then in 2020, our Common Repository Working Group just published a programming interface for curating and sharing data in the AIRR-seq, in the AIR Data Commons. And so now I want to really have a pitch here for all of you to join the AIRR Community. And our work really represents a starting point for further collaboration with the global scientific community on AIR-seq data standards. So please, again, visit our website, join our working groups, and become a member of the community. Also, please stay tuned for our next virtual meeting, which is called Zooming Into the AIRR Community, and this meeting will be held in December of 2020. More details will be coming. Also, if you have any questions at all about AIR-seq data or the AIRR Community, please feel free to reach out to us. Again, my name is Nina Luning Prak. Here's my email. And also Pam, please, our meetings committee also has a contact email right there. Next, I want to turn to describe a little bit about the AIRR Community's response to COVID-19. So our response to COVID-19 has really focused in 4 general areas. The first is that we've been working on uploading, curating, and sharing AIRR-seq data via the AIR Data Commons. So there are data from several different COVID studies that are available through the iReceptor Scientific Gateway. The website is here. You can see on this graph here on the right that the AIR Data Commons has a couple billion sequences in it, and that's since May of this year. We've been adding about 700 million AIRR sequences, which will be available probably in about a week. We're going to have the rest of these sequences up and fully available to you. So this is really a very nice resource, I think, for the community. The second thing we've been working on is tracking AIRR-seq papers and data sets on the BTCR website, and that's shown here. Third, we've of course been in active discussions about immune repertoire profiling studies and data sharing initiatives in the working groups and subcommittees. And fourth, last but not least, we've organized a virtual meeting on AIRR-seq data and studies for COVID-19, and that's this event. So without further ado, let me just provide a brief overview of today's meeting that's going to extend, of course, into tomorrow and Thursday as well. So this is a 3-day meeting. The first day today will be focused on T-cell receptor studies. The second day will focus on B-cell and antibody studies, and the third day on data sharing. In the first 2 days, the meeting will take the following format. We'll have 4 presentations on various aspects of COVID biology and immune repertoire profiling. So today we'll have presentations on how T-cell repertoire data inform our understanding of COVID-19. There will be a question and answer period after each talk. So each talk will be 23 minutes, and then there'll be a roughly 7-minute period for questions and answers. Then at the end of the 4 talks,
where are we at?
yeah? Sorry, somebody's not muted. Then there'll be a panel discussion at the end of the session with prearranged questions and time for question and answers from the attendees as well. Then in the third day, this format will be a little bit different. We'll have 3 panel discussions with prearranged questions for the panelists and time for Q&A from the attendees. The full agenda for the meeting should have been emailed to all of you, but you can also download it at the website shown here.